back, everybody. My name is Steve Schachter, and I have the privilege of being the host for the next session, which is called Clinical Stage Drugs. Yesterday, Roger Porter talked about the Valley of Death, and here we are now. We're going to be presenting 12 products that have walked through the shadow of the Valley of Death and, having feared no evil, are now in clinical stage. And so we're quite excited by that. The good news is we have 12 clinical stage products to talk about. Unfortunately, we only have 2 hours to do it. So I'm going to try to keep our speakers to their 10 minutes of allotted time. And hopefully they'll finish a bit earlier than that so we can have a question or two. So let's then proceed to our first presentation by Emil Kakas, who's CEO of Ultragenyx Pharmaceuticals, to talk about triheptanone.
Thank you. Thank you, and I want to thank the organizers and Jackie for inviting us to talk about triheptanone today. I'm the CEO of Ultragenyx. It's a company that's just, oops. Can we put it in presentation mode? Green should push it forward, but it's pushing it out. What? I am. Well, all right, one more time. There we go. All right, we're building a rare disease company that focuses on therapeutics, both biologic and small molecule, for rare diseases. We founded the company in April 2010, just up the way here across the Golden Gate in Marin County. We have 5 programs now already in phase 1, 2. 3 of them already generated positive phase 2 data. We managed financing well. We've gotten actually through an IPO, and actually at the end of Q1 had $165 million in cash, which gave us the reserves to actually drive ahead a lot of development. So I can understand the terrible situation, some of the great molecules that were here yesterday in that Valley of Death, but you can get through. We managed to get there. We're now 84 employees in Novato. Our pipeline, just to touch on, consists of biologics, including a couple for connective tissue type disorders, Karen23 for XLH and recombinant human GUS for MPS 7. Those are not in the neurological space, but on the bottom there is the triheptanone program and salicylic acid extended release. The SAR program is for hereditary inclusion body myopathy, and some of you neurologists may have patients with HIBM, and that showed positive data, 48-week data in December, and presented earlier at AAN. The triheptanone program is the one I want to focus today. It's a substrate replacement therapy that is basically an engineered triglyceride that delivers energy into the mitochondria in a way that bypasses some of the defects and restores some of the dysfunction that occurs in mitochondrial and other disorders. The fatty acid oxidation defect we won't talk about today, but are diseases that have a defect in fatty acid oxidation that result in a series of problems, including muscle problems such as rhabdomyolysis, liver and heart problems. Now, I'll talk today a little bit more about triheptanone for glucose transporter type 1 deficiency, or GLUT1 deficiency syndrome. It's, the substrate replacement concept here is in this disease is based on the fact that they are missing or inadequate levels of energy, and the idea is that this triheptanone is a way of replacing the missing substrate. So let's talk about GLUT1 first, and we'll talk about the mechanism of triheptanone. GLUT1 deficiency syndrome was originally thought to be an extremely rare disorder discovered by Dr. DeVivo, and over the last few years it's become clear that it is much more common than realized, with a rather pleiotropic set of phenotypes that are possible. Disease is all caused by a defect, an autosomal dominant defect, that is a 50% loss of the glucose transporter. If you have no transporter, you do not survive as an embryo, so all the patients have at least 50% of function. And when you don't have enough glucose, of course, the preferred substrate for the brain, you end up with seizures and epilepsy that's usually refractory or very difficult to treat with traditional AEDs. Patients often have cognitive developmental delay that can be progressive over time, and movement disorders commonly, a variety of different types of movement problems. All of this is basically due to the lack of glucose transport in the brain. In the PET scan there, just showing you some work of Dr. Pascal and Dr. DeVivo on showing the difference in glucose uptake in the brain of patients with GLUT1 versus normal. The prevalence now is thought to be 3,000 to 7,000, and this is coming from some surveys looked at idiopathic generalized epilepsy, where they sequenced patients consecutively and discovered perhaps about 1% of all idiopathic generalized epilepsy is in fact GLUT1 deficiency, undiagnosed. Another study looking at early onset opson seizures found that 10% of their clinic actually had GLUT1 deficiency syndrome as the cause of early onset opson seizures. There is a number of other phenotypes, and seizures being one of the common ones, but these patients have very complex seizure patterns. They can have multiple types of seizures in the same patient. Now, I'm not a neurologist. My father was a neurologist, so I grew up with neurology at home, but these patients have an interesting complex pattern of seizures. So the current standard of care for GLUT1 has been, there are really no currently approved specific drug therapies, and traditional anti-epileptic drugs are relatively ineffective in these patients. Many of them do take one or more AEDs, but they usually are not well controlled. But the ketogenic diet has been shown to be very effective in controlling seizures, and this involves a diet of 70–80% calories as fat. I think it's been talked about yesterday. And it introduces an alternative energy source, the concept of delivering ketone bodies, although, as was discussed yesterday, we don't really know if that's the whole story, that's part of the story. Is there an effect on transmitters, neurotransmitters, or other things beyond energy? I think those are some pieces that are not known. But the concept is ketone bodies get across the blood-brain barrier, and so that block to glucose is bypassed. While it's been very good in seizures, it's clearly not been optimal in developmental delay or cognitive problems or in motor dysfunction, and therefore there's clearly room for improvement beyond that. And KD compliance can be difficult. It's an extreme diet, and there are other versions of modified Atkins and other things that people are looking at, but clearly it's a demanding, and there may be problems with a high-fat diet, and a number of complications of that diet can also be have been described. So the question now, what is another way to do it? And triheptanone has an alternative strategy. Triheptanone is a triglyceride of C7 fatty acids. So these are odd-chain fatty acids. Your body has even-chain fatty acids, usually C16, C18. These are C7, so they're medium chain and they're odd. And that causes something very important in how they get metabolized in the body. What happens with triheptanone is once it gets into the gut and into the liver, it basically gets broken down into heptanoate, and the liver will further metabolize it into 4 and 5 carbon ketone bodies, regardless of whether you're on a ketogenic regimen or not. The medium chain will get broken down specifically, and so they make ketone bodies regardless of whether you're fed or fasted. The heptanoate will be released to the bloodstream as well as 4 and 5 carbon ketone bodies. The heptanoate itself actually can diffuse directly into the brain, and the ketone bodies also get across via the MCT1 transporter. So really, triheptanone provides 5 different energy substrates to the brain. And what's been studied in the mouse model of this is, in fact, that this can actually help improve the metabolism, both in restoring the function of the mitochondria, but in actually allowing gluconeogenesis in the brain as well, and I'll show you that in a moment. So when you look at how triheptanone gets metabolized, it basically gets transports across the mitochondrial membrane by diffusion itself. The medium chain enzymes then clip it into acetyl-CoA and propionyl-CoA, two compounds that enter the TCA cycle. Now, acetyl-CoA is what you normally get from fat. But the unusual piece is that the odd chain leaves you with a 3-carbon unit, which allows you to recharge the Krebs cycle. The Krebs cycle gets deficient in hypoglycemic disorders because the gluconeogenesis process drains the Krebs cycle of intermediates, and the need for neurotransmitters will do that as well. And so providing the 3-carbon unit allows you to recharge the Krebs cycle, and as you can see there through the malate cycle, allows you to generate glucose in the brain or anywhere else. 1 molecule of triheptanone actually can be converted then to 2 molecules of glucose. Fat normally cannot be converted to net glucose, but triheptanone can. So the gluconeogenesis component, as well as recharging the Krebs cycle, are part of the mechanisms behind triheptanone. And I won't have time to go through all the details. This is a very nice article by Marin Valencia, 2013, that showed through a triple metabolic labeling that, in fact, the heptanoate both is going to the liver, getting converted to glucose, as well as other substrates, ketone bodies. And the ketone bodies, as well as heptanoate directly, are getting to the brain, and that gluconeogenesis is going on in the brain, including the production of glutamate and glutamine. And the glutamine production suggests that the glia are actually dominant in the metabolism, but there is clearly C13 glucose getting taken up by the neurons as well. Some very nice work showing how heptanoate might be able to fix the problems in GLUT1. Now, There's already been some data previously not published but described in a few patients with GLUT1 that showed a significant improvement in seizures and cognitive function. Those anecdotals have not been published yet, but we're hoping to see them soon. We're conducting a randomized control study of GLUT1 patients. These are patients who are failing the ketogenic diet and cannot have breakthrough seizures. We're not asking people on ketogenic diet to be taken off. If they're on it, they're stable, they should stay on it. But if they can't do it and they're failing and they have seizures, they can enter the study. If they meet the seizure baseline rate of 4 per month, they're randomized then tryptanoid or control oil. They're continued then for 8 weeks, and the seizure baseline rate is compared. They're also looking at cognitive function as well as motor function in the study, and then they go on to an open label. That study's up and operating. If you have patients that might be interested, please let us know. We're also supporting a number of investigator programs globally, looking at combination of tryptanoid with ketogenic diet in GLUT1 patients, looking at motor dysfunction issues and the other symptoms and manifestations of GLUT1 deficiency syndrome. There's also some work going on in refractory epilepsy. So we're doing support for these programs. If you have other interesting ideas, we're certainly open to looking at those. One last piece, GLUT1 diagnosis. We think it's very much underdiagnosed, and not enough neurologists need to be paying attention to it. The diagnosis of low glucose in the CSF is certainly what Dr. DeVivo would recommend you do. But in addition, you need to be looking for and doing the sequencing, complete sequencing, and we're now establishing a free sequencing plan with Medical Neurogenics, which can help, and we're going to be rolling that out shortly. That will help us diagnose patients who have refractory seizures that have turned out to be GLUT1 and help better understand the GLUT1 population. And from the initiatives I've been hearing today of the epilepsy group, it seems like we would need to get connected up and make sure we're working closely with those existing initiatives on identifying, understanding GLUT1. That's it from me. Thank you for listening.
Thank you. Perhaps time for one question if somebody wants to approach a microphone. If not, given the complication of the cognitive delay, when, in your opinion, would be the ideal time to start therapy in a patient with this disorder?
It's definitely early. I mean, I think the data already Dr. DeVivo has with KD and treatment is, early treatment is really essential. And this is why we need to be thinking about this in young children with new onset seizures, infants essentially. They need to be thinking about this diagnosis and getting on it because early treatment can have an important impact.
Okay, thank you. Thank you very much. Our next speaker is Dr. James Casella from Alexza Pharmaceuticals to talk to us about inhaled Alprazolam.
Good afternoon. Thank you, everybody. And I apologize in advance because I have the throat thing that's going around. So, I'd like to thank the organizers and Jackie in particular for inviting us back this year to talk a little bit about our inhaled Alprazolam program. We are looking at acute repetitive seizures. I think it's preaching to the choir here, but basically these are seizures or seizure clusters that occur in patients with epilepsy. They're not typical to their usual seizure pattern, and these are what you can be considered to be breakthrough seizures. What we've looked at in the literature is, and for treatment options, benzodiazepine seems to be the drug class of choice. There's only one therapy available today for the treatment of acute repetitive seizures, and that is with a rectal diazepam gel, Diastat. It is approved, but as you look at the package insert, you can see that there are features of this route administration and the drug itself that may be less than optimal for this patient population. One of the things that we've noticed in the literature is that there are a number of drugs under investigation for the treatment of acute repetitive seizures, all benzodiazepines, primarily through the intranasal route of administration. Those drugs include diazepam, midazolam, clonazepam, lorazepam. So we're looking at Alprazolam. But before we get to Alprazolam, I'd like to give you a little bit of background on Alexza and what we do and what our technology is. So we've developed an inhalation drug delivery system called Staccato system. It's a very simple system that takes solid drug and delivers it to the deep lung. The purpose is not to treat the lung, it's to deliver drug into the bloodstream. So we've developed a system that is a single breath actuated system that delivers drug. It takes very little effort by the patient. It is very intuitive to use, and unlike other types of inhalers, there's no coordination required because you simply breathe through the device in order to deliver the drug. We take excipient-free drug. So in this case, it's pure Alprazolam. We spray coat it onto what we call a heat package, and when the patient breathes through the device, aerosol is generated in situ. So we're transforming a solid drug into a fine aerosol particle. The reason we're doing this is we're creating aerosol particles that are optimal for deep lung delivery so that we can get that drug, those drug particles directly into the bloodstream via the lung. And this is via the deep lung. With that, we have characteristics with all the products that we have using the Staccato system where we have IV-like pharmacokinetics. We have rapid absorption into the bloodstream, and we have very high bioavailability of the dose that comes out. We actually have one product that is approved in the US and in Europe using the technology. It's called Adasuve, and it's an inhaled form of loxapine, which is used in treating acutely agitated patients who have schizophrenia or bipolar disorder. So it's a product that's used in an emergent care situation. Because the technology is really important here in delivery of the drug, I wanted to go into a few of the details of how it actually works. So if you look at what we see here on the screen, there are a couple of various components to it. There are some electronics and a battery in it. Mostly the electronics are involved in sensing the patient's breath. So when the patient is breathing through the device, there is a trigger in there that when it reaches a certain airflow, which is not much more about what we're doing right now, sitting at rest, it will cause a electrical circuit to actuate and start the reaction. The drug is coated on what we call a heat package. It's coated by basically spray coating the pure Alprazolam on there. Solvents evaporate off, and we're left with a solid drug film. And when the patient breathes through the device and actuates it, the heat package initiates a heating reaction. So we're getting the drug on the surface of that heat package up to about 350 degrees Celsius. It rapidly vaporizes that drug, forms the particles that we know are important, the size are important for getting into the deep lung, and those are pure Alprazolam particles. If you look at these pictures, which are taken from some high-speed photography of the actual vaporization process, you'll see in the upper left-hand corner at time 0 before the patient has initiated the reaction, there's drug coated on the stainless steel substrate. 30 milliseconds after the patient has breathed through the device, you can see the cloud starting to form. That cloud are thousands of aerosol particles that are 1 to 3 microns in size. By 50 milliseconds, you can see that most of that drug has been vaporized off and is in the airstream that the patient has generated. And by 200 milliseconds into the process, that patient has totally vaporized that drug, and that drug is now in their deep lung. Because we have such rapid generation of the aerosol, and we have particles that are optimal for getting it into the deep lung, we have rapid delivery into the bloodstream. So this is data now from our phase 1 study and from our phase 2 study in non-epilepsy patients, where we show that the plasma time concentration over 2 different doses. The important point to note here is that when you look at the time scale, you're seeing that we have a rise to maximum concentration within 2 minutes. That is dose independent. So this is a hallmark of the technology. We not only see this with Alprazolam, but we see this with all of our other compounds as well. So again, by generating these types of aerosol particles, we get the drug into the bloodstream very quickly. Just for a perspective, and just as a put it in relationship to the other drugs that are being developed for ARS, you can see down in the lower corner, the staccato delivery of Alprazolam produces a maximum concentration within 2 minutes. Just to look at some of the other drugs given by intranasal administration, again, these are from literature papers, and we can provide the references if you need them, but they're all out there. Midazolam, Lorazepam, diazepam, clonazepam, with the studies that are available in the literature, have a variety of time to maximum concentrations. As we noted earlier on our first slide, if you look at the rectal gel, of course, the time to maximum concentration in the bloodstream is an hour and a half. So for people that are having repetitive seizures, we think it's very important to have rapid onset of effect. And here we're clearly showing that we can achieve rapid concentrations in the bloodstream. We know from studies in phase 1 and in phase 2 with inhaled alprazolam that looking at pharmacodynamic measures such as sedation using visual analog scales, we see maximum effects within 2 minutes. So the PK seems to be following, or the PD seems to be following the PK. So just as a final slide on the update of where we are with staccato alprazolam for acute repetitive seizures, we have an active IND. We've studied we've had 3 different studies to date with about 100 subjects and patients studied, all showing that the drug is safe and well tolerated. We are planning, in the late stages of planning, a proof of concept study in epilepsy patients. We're going to use the intermittent photostimulation model to determine an active dose range of alprazolam. We're initially going to look at 3 different doses. We'll establish the photosensitivity range and then look at the suppression and abolishment of that effect with the various doses of inhaled alprazolam. And we expect to get that study started probably within the next quarter. And I'd like to take any questions that you might have.
Thank you very much. Any questions from the audience? Jim.
It wasn't clear if this device is intended to be a single dose or multiple dose product.
So the device that we have in development is a single dose product, as is our commercial product that we have with Adasuve. We do have a multiple dose product capability, and we have another product line, but our intention here with this would be a single dose product.
And it's hard to tell from the component parts. Would you anticipate that circuitry and component parts would represent a substantial cost?
There's costs associated with it, but
Sure.
electronics are very cheap these days. So I think we can be competitive with what's out there for current treatment, for sure.
Thank you. Jim.
Just, I'm trying to understand the sort of safety profile. So if you're getting Cmaxes and Tmaxes that look like IV, generally patients are dosed IV in a hospital setting because of the rapid sedation. So can you help me understand how somebody in an ambulatory setting would be using this and not put themselves at safety, at risk? Let's say they're driving or walking or doing something, operating, I don't know.
So obviously the concept here is that for acute repetitive seizures, we will be treating post first seizure. So the patient will be using it. They've had a seizure, so they probably aren't going to be driving. One of the things that we're looking at and what we plan on doing with our dose ranging study is to look at the effect on the EEG pattern versus the sedative profile of alprazolam. I mean, one of the reasons why we chose alprazolam is that while all benzodiazepines are sedating, we see less sedation in healthy volunteers. We see less sedation we were in a panic population before. So that's one of the things that we really have to do in the first study is to look at the changes on the EEG pattern versus what we'll be measuring, which will be sedative parameters such as VAS scores and things like that.
Great, thank you.
I apologize, but we're going to have to cut it here, and you'll be available afterwards for further questions. Thank you.
Thanks.
Great. Our next speaker is Alice Mead, who is with GW Pharmaceuticals to talk to us about Epidiolex in pediatric epilepsy.
Good morning, and many thanks to Jackie and the other organizers for giving us a chance to update you on our progress. So I don't need to tell you that there's a great deal of public interest in the use of CBD in the treatment of childhood epilepsy. It seems like daily the newspapers are presenting the cases of desperate families that are trying to change their state laws or travel to Colorado to obtain preparations. Dr. Sanjay Gupta profiled a particularly dramatic case in his CNN program, and an excellent survey was published by Drs. Jacobson and Brenda Porter, and I recommend that to your reading. So there's a growing body of anecdotal evidence, but there are no rigorous clinical trials or clinical evidence, and that is what GW intends to provide to physicians and to patients through our development program. So we are not just a Sanjay Gupta opportunist. We have been thinking about CBD for a very long time. We were founded 16 years ago in the UK to develop a range of prescription medications that are derived from the cannabis plant. And way back then, we thought that CBD had significant therapeutic potential. So in fact, it forms half of the principal cannabinoids of our first product, Sativex. And because of the development of Sativex, we have amassed a large body of preclinical and clinical safety data. So that is already in place. About 7 years ago, we began more intensively to conduct preclinical research into CBD and other non-THC cannabinoids in a variety of medical conditions in a number of models. And in epilepsy, 2 product candidates emerged, CBD and CBDV. But actually, fast forward just a bit, US physicians urged us to take the CBD and develop a formulation for children and to conduct a US development program because they were increasingly concerned that their patients were being exposed to different vernacular preparations of uncertain quality and safety. So hence Epidiolex was born, which is more than 98% pure CBD in a liquid formulation. We are also pursuing the other candidate, CBDV, which is a structurally related molecule with a different side chain. It shares some mechanisms with CBDV, but it has different safety profiles and efficacy profiles in different mouse models. It shares, as I said, some mechanisms, but it hits different ion channels and different enzymes. So we have already completed a phase 1 in CBDV, and we intend to begin a phase 2 sometime next year. So the problem with most of the CBD products in the US are that the composition, the quality, the consistency, and the safety are unknown and quite questionable. And increasingly, there are reports that many of these products may be contaminated with heavy metals and pesticides and contain significant levels of THC. GW conducts all of its cultivation and manufacture under GMP in accordance with strict standard operating procedures and quality control processes. We conduct all of our cultivation by clones in organic medium in glass houses in the UK where every aspect of the cultivation cycle, which is 12 weeks, is computer controlled. We extract the active ingredients, and in the case of the CBD extract, we put that extract through additional purification processes to remove the THC. We remove the THC because we think that the very young brain is different from the adult brain. The very young brain, if constantly or chronically stimulated by THC, that may lead to untoward, unintended, and just a whole variety of consequences that we don't fully understand. But we do know that the very young brain is undergoing rapid neuronal development, and there's a growing body of evidence that THC can inhibit and distort that neuronal growth. There is also evidence that THC in some models and in some animal species is proconvulsant. So we remove the THC. We think cannabinoids offer an exciting new class of AEDs. The endocannabinoid receptor system, or ECS, is sometimes called a supermodulatory homeostatic mechanism that achieves balance in the body by a variety of mechanisms. Cannabinoids don't have a single target or a single mechanism of action. One possible target would be that the ECS can inhibit the release of excitatory neurotransmitters. And CBD modulates the ECS and other receptor systems and non-receptor mechanisms. Inflammation is important in the development of epilepsy. There are no medications that currently target the inflammatory processes, but CBD is a potent antioxidant and anti-inflammatory molecule. It also inhibits the release of pro-inflammatory cytokines. It's clear that ion channels play a role in the development of epilepsy, and CBD and other cannabinoids modulate the flow of cations and in particular intracellular levels of calcium. The GW program is moving quickly. We've secured orphan designation for Dravet syndrome and Lennox-Gastaut syndrome for Epidiolex, and just today we announced that we had secured fast-track designation for Epidiolex. We will also seek breakthrough designation and other accelerated pathways. We have two prongs to our program. One is the GW-sponsored placebo-controlled clinical trial program, and the other is a series of physician-sponsored INDs under FDA's expanded access regulations. Those began with individual patient INDs, then 5 intermediate-size INDs were approved for 125 patients. Now 300 patients have been approved in 12 sites to receive treatment with Epidiolex for a variety of epilepsy conditions. The first 2 of the intermediate-sized sites began in January, and we expect to release data at mid-year, which is almost here, so stay tuned for that. The GW-sponsored program is, as I said, moving well. We've opened an IND with FDA with a phase 2/3 protocol in Dravet, and we intend to begin the first part, which is a PK safety study in the second half of this year, with pivotal studies in Dravet and LGS to begin in early 2015. It should be noted that CBD is a Schedule I substance under the Federal Controlled Substances Act. It doesn't matter whether it comes from hemp, a laboratory, or a THC-containing cannabis strain. And so each research site must have a DEA Schedule I research registration. The expanded access program, the physician INDs, are also going well. There are over 600 weeks of exposure and a range of continuous exposure of up to 60 weeks. Only 1 patient has been withdrawn for lack of efficacy, and this slide gives you the range of epilepsy conditions that are being treated in those expanded access INDs. We think that this will generate a very interesting data set, which may guide us in future development programs, and we hope will be of interest to the scientific community. This just gives you an overview of the timelines of our program. As I said, we expect to begin the first part of a phase 2/3 protocol in the second half of this year in Dravet, with pivotal studies in Dravet and LGS to begin in early 2015. The expanded access INDs will continue until Epidiolex is approved by FDA, and we will be making data communications at regular intervals. Finally, CBDV, having finished phase 1, we will open an IND with FDA with a phase 2 protocol later this year and begin that phase 2 study in early 2015. Thank you very much for your attention.
Thank you. So we have 2 minutes for questions. First, Dr. Katz.
Yeah, do you anticipate any problems with enrollment in your controlled trials for Lennox-Gastaut and Dravet, given that you have expanded access programs and some of the patients in those programs have those conditions?
Some do, but there are a very large number that actually don't have Dravet or LGS. And as the new expanded access programs open, they're trying to reserve some of their LGS and Dravet for our clinical trial program. We collaborate, so you know they don't operate completely independently.
Any other questions? Can you tell us about the bioavailability of the liquid formulation?
Well, you know, bioavailability, I may not be the best person. I wish that Dr. Guy could be here, but he was detained in the UK, and I'm an attorney in the Sea of Scientists. But you know, the oral formulation is probably not as bioavailable as an inhaled or an IV formulation, but it's still for children. For chronic conditions, this would be better.
Any other questions? If not, thank you very much.
Thank you.
Next speaker, Dr. Stephen Collins, CEO and President of Insero Health, to talk to us about Huperzine.
Well, thank you very much. It's quite an honor to be here. It's nice to see such a large group of people with interest in epilepsy. Insero Health's mission is to develop drugs for conditions which don't currently have any effective therapies, and in particular, conditions which have catastrophic endpoints. So we have a philosophy of trying to help those who need the help the most, and the way we're trying to pull that forward is by taking traditional medicines found around the world and improving them. The benefit of this, of course, is many of these medications have hundreds of years of safety data. And as opposed to trying to develop medications on a new target where you unfortunately find numerous side effects for patients, we believe this will help decrease the chance for safety problems. So just briefly, I'll go to an overview of the company. Our focus, as I say, is in rare and catastrophic diseases, so-called orphan diseases. Our first indication for our lead program will be in Dravet syndrome. The lead compound is a synthetic form of a medication which has been used in China for hundreds of years, primarily for the elderly to help cognition as well as in pain and inflammation. The company is a clinical stage company. This asset has been in phase 1 studies in adults with refractory CPS. And our next stage will be to develop a pediatric friendly formulation which will be used for children with Dravet. We have a team that's developed multiple medications in this space before, and we have good IP and other protections. Clearly, what we'd like to do is move this along and partner it with a company that would be able to take it all the way forward. Just briefly about the company, the management team is the same team that developed in the United States vigabatrin for infantile spasms, clobazam for Lennox-Gastaut, Depacon, Depakote ER, et cetera. Scientific Advisory Board has some unknown people like the guy to my left. Stephen Schachter is the chairman and the scientific founder of the company. That was a joke, Steve. Roger Porter, ex-president of AES, and obviously well known to this crowd, as well as Steve White from the ASP. Warren Lambert and Sam Reich are the 2 other board members in addition to myself. Well, the highlights, I'm not going to go through all of this. The bottom line is this molecule has been around in China for hundreds of years through the ASP program, and I'll show those data in a minute. It has shown tremendous efficacy. The potency is one of the most potent drugs the ASP has ever screened. As an example, it's 57 times more potent than Keppra in the 6 Hertz model. The differentiator here is that this drug aids in cognition, and as those of us who are either patients or physicians who work with patients with epilepsy know, the majority of the medications, in fact, are anything but cognitive enhancing. So we have a way of manufacturing this material from a completely synthetic method, and we have the intellectual protection based on that synthetic method. We've already demonstrated that we're able to manufacture up to 50 grams of this highly potent compound, and so we're poised, ready to make an extended release pediatric formulation, juvenile tox studies, and then a second IND, this time for Dravet. We hope that will happen in 2014. Excuse me, late 2014, early 2015. This is some of that, quote, ASP data, and what you can see in the green is that at the classic step of evaluation, 32 milliamps, INS-001, which is Huperzine, is far more potent than Keppra. And in fact, Keppra has basically no efficacy at the more impressive stimulation of 44 milliamps, and yet INS-001 is still quite potent in a range which humans have actually experienced before, a dose range. So as I said before, our lead indication is Dravet. I hardly need to discuss Dravet with this audience. Clearly, huge unmet need, and while I hope that many of the therapies people are talking about here will help some children with Dravet, the unfortunate reality is it's highly unlikely any one medication will help all the kids with Dravet. So for a bunch of complicated science reasons I won't get into, we had a belief that Huperzine would help in Dravet, and so with the help of Dr. Andrew Askai, the very generous help of Dr. Askai at Emory University, using a genetic model of SCN1A, that is Dravet, we were able to show that at very low doses, we were able to obtain 100% control of seizures. Now, it's a little tough to see, but all the stars at the bottom are animals that have no seizures. And so the majority of animals actually had no seizures in a dose range if you scale it up to humans, which would be quite practicable. So the summary is this is a clinical stage company with Harvard roots, ergo Ivy League. We have an experienced science and development team to pull it forward. We have a validated lead compound that is over 400 publications on the pharmacology, the preclinical safety, and hundreds of years of safe use in China. It is a novel mechanism, very clearly one that can be used for epilepsy, pain, and inflammation, but has not yet been. We have IP around manufacturing methods, as well as formulation, et cetera, as well as a pending orphan status. In summary, Insero Health is poised to take this asset into kids with Dravet as soon as we can. Thank you.
Thank you. We have time for a couple questions. from the audience?
Just want to send checks in, that's okay too.
Andy?
Steve, this isn't meant to be a nasty question, but I'm curious. Is there any evidence from the Chinese experience that kids with epileptic encephalopathies, or even Dravet more specifically, have had a benefit?
No, it's a very good question. You know, the Chinese literature speaks in general terms about this, but there haven't actually been studies. We did our synthetic manufacturing with WuXi, one of the premier manufacturing companies in China that supplies for Glaxo, BMS, et cetera. And we have a Chinese partner that we're in diligence with to be able to potentially go forward in China. Anecdotally, we've spoken to some docs in China and say, oh yeah, I've used it. The reason it probably is not getting used is one I didn't get into. This is very bioavailable. If you take one of the capsules now, you'll get a serum peak almost instantly. And at very high dose, you'll have nausea and may even vomit. That's why this needs to be an extended release. You could go to GNC and buy this stuff, but the reason you probably wouldn't use it in your child is that you'd have to be giving it every 2, 3, 4 hours. It's very rapidly cleared through the body, and that's probably why. Now, in cognition in China, it's used twice a day. It probably would be much better if it was an extended release formulation. Thanks.
Another question, please?
Can you speak to potential utility in seizure types outside of Dravet, and then conversely, your thoughts about your IP position in those seizure types?
So to take the last question first, we have a method of use patent around seizures and pain that's worldwide. To get to the other question, the ASP screen would indicate this should work in refractory complex partial seizures. It also works in the MES model that would help explain potentially to help progression of seizures, that is to say spread of an individual seizure. It does not work in PTZ and some others, so my guess is this is not going to be a drug like valproic acid in which you get effect on absence seizures, drop seizures, tonic seizures, etc. Drug-drug interactions, glad you asked. Probably not going to have significant interactions.
That was my next question.
Probably not going to have significant interactions with stiripentol, clobazam, et cetera. Can't exactly answer that right now. And so add-on trials in Dravet would obviously be adding on to those medications. Looks like it's going to be okay. Thank you.
Great. Thank you. Thank you, Steve.
Thanks, Steve.
Next presentation will be from Dr. Gail Farfel from Marinus Pharmaceuticals to talk to us about Ganaxolone.
Thank you, and I'd like to thank the organizers for welcoming us back again this year. Let me make sure I can. Right. I'm going to focus on what's new with Marinus since the last time I presented 2 years ago. Most importantly, we have a new CEO, Chris Cashman, who I think I saw in the back somewhere, and a new CFO, Ed Smith, and all now located, we have a new office in downtown New Haven, Connecticut. We have, since the last time I presented, two of our patents in the US and Japan, Australia, New Zealand, a few other countries they're rolling out, have been issued for covering our nanoparticulate composition, which is very important, as you may remember from previous talks, in the bioavailability and managing the fed-fasted effect for this preparation to make it into a compound that could be approvable with the right studies. We also have issued our synthesis patent, which is also important, and so this constellation of patents will provide protection for ganaxolone until 2026 or later, which has really reinvigorated the company. For the remainder of the program, I'll talk mostly about our epilepsy development program. However, we also have proof of concept studies ongoing. We have one in a pediatric population, 6 to 17, in Fragile X syndrome. And then we have a proof of concept study in PTSD that is nearing completion. So it has been very gratifying throughout this meeting to hear the talk about GABA. There wasn't much talk about GABA for a few years, and I felt pretty alone. Especially the information about the interneuron therapy this morning was very exciting. How does ganaxolone work at GABA? It's part of the class of neurosteroids, the positive allosteric modulators, which are metabolites of cholesterol and by way of progesterone. What's important about allopregnanolone is that it is metabolized from progesterone and can be back converted to an agent that can be active at the nuclear progesterone receptor. So ganaxolone was synthesized originally by consensus in the 1990s with a methyl group at the 3-carbon position, and that prevents the conversion to an entity that is active at nuclear progesterone receptors, and thus we are developing it for chronic use. So just to go a little bit into the chemistry of neurosteroids and these extrasynaptic GABA receptors, I think we've touched on them a few times already this meeting. The synaptic GABA receptors are the ones, the darker blue, and then the class of extrasynaptic receptors are the lighter blue that are postsynaptic outside the cleft, appear to differentiate in terms of their availability, particularly in certain seizure syndromes. And with the lipophilicity of allopregnanolone and by extension ganaxolone, there is a diffusion into the postsynaptic membrane that provides this constant low-level tonal modulation that provides a GABA signaling that is different mechanistically from the synaptic signaling. And so we believe is the basis for the difference, particularly with the benzodiazepines, in terms of the lack of observed tolerance that we have seen. In terms of development, ganaxolone, as many of you know, has been in development for a number of years now. We have 4 open INDs in both adult and pediatric indications. We've completed our preclinical IND-enabling and preclinical testing package with the only exception of the 2-year carcinogenicity studies. And what is most interesting about the drug is the clean safety profile that we see in this testing. Safety pharmacology, we don't see any end organ toxicity that's been identified. The genotox studies, similar. The reproductive toxicity studies were clean. There was no findings in embryo-fetal development, no postnatal findings, no fertility findings. And then the chronic toxicity that's been completed to date, the 1-year dog studies were also clean. So very, very encouraging profile for a drug that could be used to treat adult populations as well as pediatric populations. It is metabolized by CYP3A4. It's a common target that we have all managed, I think at one time in our careers or another. Ganaxolone in a midazolam probe study was shown not to be either an inducer or an inhibitor itself, but of course interacting with common CYP3A4 drugs, and the most obvious for an epilepsy meeting are carbamazepine and phenytoin. There would be changes there in ganaxolone levels. Now, the next 3 slides are data that I have presented previously. I want to remind you of what we have accomplished in the program. I am going to go through them quickly so that I can talk about our new study. And I understand they're not available on the app yet, but they will be up later today. There are 3 studies to discuss. A phase 2 study in drug-resistant partial onset seizures in adults. That was presented last time. 147 subjects, 98 were on ganaxolone at 1,500 milligrams a day. And there was a significant difference detected between the mean seizure frequency change in the ganaxolone patients relative to the placebo patients. The mean difference was almost 20%. The median difference was 16%. And then the data in the figure on the lower right represent the 50% responder rate. We did have an open label extension to this study, excuse me, and these data were presented at AES a few years back, and the findings from the open label extension supported these findings, and the mean changes in the group that went out to 26 weeks were approximately 25% improvement, with almost 40% improvement at the end of 1 year for those subjects who were still in the study. The safety profile from the study is shown in this slide. And I'm not going to try to work with the pointer, but dizziness, fatigue, and somnolence were the only adverse effects that were noted at an incidence over 5% that were higher in the ganaxolone group compared to the placebo group. Everything else in terms of AEs was actually the same or higher in the placebo-treated patients. Notable in this study and the open label extension and the program to date is that we haven't seen any findings on ECGs at all. the occasional individual, but nothing that indicates a trend, nothing that is population-wide, and nothing that's persistent. We haven't seen changes in hematology. We haven't seen changes in vital signs. The clinical picture really does support what we saw in the early pharmacology studies. These are data from a publication in Epilepsy from 2000 that many of you are probably familiar with. This was the pre-surgical model, so it is, in a sense, a proof of concept study of ganaxolone monotherapy. These were 52 subjects, half treated with ganaxolone 1,875 milligrams a day, and half treated with placebo. And the Kaplan-Meier survival curve, the ganaxolone is the dark line on the top, excuse me, shows the subjects who survived the 8-day treatment period without meeting the criteria for discontinuation due to emergence of seizures at the end of the period. So with all that, it gets me to where we are now. And after talking with many of you after this meeting and at many other meetings, the question was, well, with a profile such as yours, would you get enhancement of efficacy if you ran a higher dose of ganaxolone? And so these are data from a PK study that we ran in the past year, looking at 3 dose levels, 1,200 milligrams a day, 1,600 milligrams a day, and 2,000 milligrams a day, given as divided dosing or BID. And subjects were inpatient, healthy, normal volunteers. They were dosed for 3 days at each level, and what you're looking at, the dots are the trough levels, and then the little spikes are the full PK curve at each of those dose levels. And so what we see is between the 1,200 milligram a day dose, this is with our BID capsule, so it's a new formulation. At 1,200 milligrams, going from 12 to 1,600, there is, it's a logarithmic graph, so it's a little bit hard to see, but there is essentially a conservation of the dose proportionality. But when you go from 1,600 milligrams a day to 1,800 milligrams a day, you do lose that proportionality. It's about a 25% increase with a concomitant increase in trough levels of 10%. So that gives us a sense with this formulation of where we need to look in order to increase the dose in a clinical study. The last slide is the design of the study is on clinicaltrials.gov. It's an ongoing trial. Cohort 1 is fully enrolled, and it is a comparison of 50 individuals, adults with partial onset seizure, drug resistant, that are dosed for 4 weeks at the 1,200 milligram level and then 4 weeks at the 1,800 milligram level. Those individuals are almost completed with their treatment, and the safety profile, looking at it from blinded reviews, does appear to be consistent at both dose levels with the safety that we have observed in other aspects of the program. And then we are currently also enrolling in the second cohort, which is a 12-week maintenance phase of the 1,800 milligram, which we hope when completed can be part of our registration package. So with that, I
Good, thank you very much.
take questions.
and 100 people randomized, again in an adequate and well-controlled trial design. Treatment was once a day. Titration was gentle, 2-week intervals, 50 milligrams, 100 milligrams, 150, and 200. We didn't force people to titrate up to 200. We let them settle out at the dose that was tolerable and seemed effective to them. And if they were intolerant at a dose, we would allow them to drop back 1 dose level. The first clue comes from the subject disposition data. 113 people randomized to YKP 3089, 109 to placebo. In terms of discontinuations for any reason from the double-blind trial, results between placebo and drug are the same. In terms of completion rates in the double-blind portion of the trial, results are the same. In terms of discontinuations for adverse events between drug and placebo, results are the same. Of those, not every country allowed us to have an open-label extension trial. Of the countries where we were allowed to have the open-label extension trial, about, I think, 92 to 93% of patients chose to go on to open-label. Most of them are still with us now. In terms of demographics and baseline characteristics, basically randomization worked for gender, race, age, concomitant medications, and baseline seizure frequency with no statistical difference between any of them. As you know, the primary outcome variable in these trials is typically your percent reduction, your median percent reduction in seizure rate. In the current era, placebo tends to run around 20%, plus and minus a little bit. Typical effective anticonvulsants run around 35% in their response. Excellent drugs, from my point of view, sort of best-in-class drugs, I think Keppra, Vimpat as an example, tend to run around 40%. In comparison to placebo, normal, 21.5%. SKP, 3089, 55.6% reduction in seizures, significant at the p.0001 level. So the next question is, so how robust is it? How does it affect the different subtypes of seizures? Against simple partial motor seizures, we were able, with a mere 30 patients basically in each of the groups having those types of seizures, we were able to achieve statistical significance in part because the effect size is so huge, 76.3% versus 27.8%. In complex partial seizures, which are of course the dominant seizure that you see, since that's what the trial was intended to recruit, again the results 55.6% versus 21.1%, significant at the p.001 level. In secondarily generalized tonic-clonic seizures, again we achieved statistical significance with a 77% reduction in seizures. The second way you look at these drugs is by the European criteria. How many people had at least a 50% reduction in seizures? By that standard, we were statistically significant at the .0001 level with a 50.2% of patients having at least a 50% reduction. We started thinking about this. Actually, we were thinking about this before the trial concluded. We were thinking, what would be something compelling? What would be something dramatic? We concluded, well, a 90% reduction in seizures would definitely have to get people's attention. So that was one of the measures. Fully a third of the patients, statistically significant at a .0001 level, had a 90% or more reduction in seizures. But we were also thinking, what's the point of this? What's the real goal? I think we've heard it referred to again and again in this meeting. What do you want to do? You want to eliminate the seizures entirely so that people can go about living normal lives. The difference between 1 seizure and 0 seizures is the difference between being able to drive and run your life and being impaired. So we looked at the number of patients with 100% cessation of seizure during the maintenance period when they had achieved their full dosage. 27.5% of patients achieved that. Some of these patients came in with 15 seizures a month and went to 0. often on the first dose at the 50 milligram dose forward. Separate, again, statistically significant at the 0.0003 level. In terms of serious adverse events, it is unsurprising that for placebo, the serious adverse events were they had seizures, right? Status epilepticus, or a major attack of seizures. For the drug, we had one patient with a UTI, and another patient who on their very first dose got some redness in their palms and feet, were taken to the hospital. It was thought to be probably a hypersensitivity reaction, given steroids, sent home, did fine. In terms of adverse events, the adverse events are adverse events familiar to everyone who's ever treated epilepsy. Somnolence, dizziness, nausea, fatigue, right? They come on from the first dose, from 50 milligrams. They typically blow over, as these things tend to do in the course of a few weeks in most patients. I might note that it's interesting that in the normal healthy volunteers treated with this drug, we treat people to 500 milligrams without significant incidence of these adverse events. It's very clear that when someone's already on 3 medicines and then you pile on a 4th, you're going to get a higher rate of adverse events than you do from the drug alone. And indeed, in our open label trial, a number of the investigators started discontinuing other drugs because we allowed them to then pump our dose of drug up to 400 milligrams. Our conclusions: YKP-3809 is a new chemical entity, has multiple mechanisms of action, selected by, as I spoke about yesterday, pathophysiological screening. It's been assessed in an international, multi-center, double-blind, randomized, placebo-controlled trial as adjunctive therapy in partial complex epilepsy. I might note that we saw the same direction of results in every country in the world. It's not driven by just one country. Once a day, well tolerated, a significant improvement in every measure of seizures that we were able to think of before the trial concluded, including the European and US criteria. Most interestingly to us, notably present was a dramatic increases, 90% or more decreases in seizures, and 100% reduction of seizures during the maintenance period. Adverse events are AED adverse events. Not a lot to say there. We think this is a promising new therapeutics. You can make your own decision whether this begins to change the game. I would say, having registered or worked in programs that led to the registration of a lot of drugs in my life, you have to do it twice before you can truly believe. But at this point, we're pleased. Thank you very much. I appreciate your time and attention.
Dr. Katz, please.
Yeah, 2 questions. The best way to distinguish yourself from another drug is to compare yourself directly to another drug. So I'm wondering whether or not in your plan you intend to do a study looking at that. The other thing is, and so that's the first question. Second question is, do you intend to study a higher dose?
Yes. We're deeply into our phase 2b trial. I decided to allow the dose to increase up to 400 milligrams, again, as tolerated, because I want to drive that % elimination of seizures up to the highest level possible. I mean, I'd like to see that get to 30 or 35 or 40%, because that makes such a huge difference. So we're trying a higher dose. I might note, though, that about 2/3 of the patients got up to the 200 milligram dose. About 1/3 finished out at 150 or 100 and a few at 50. And they all did well as well. So we're also checking a 100 milligram dose, because that may be sufficient for a lot of patients. In terms of comparator trials, yeah, if we're able to replicate this data, it's simply a question of who do we attack, right? Who is the drug, preferably the one with the best price, the one that's the most efficacious drug in the market? Who do we take as a comparator? This is almost a requirement for Europe, from my point of view, for pricing authorities. But I think it's also a useful thing to do, because from our point of view, differentiation is not just about being clinically more useful. Differentiation is something you have to demonstrate to win in the marketplace. We think that head-on trials are a great way for us to do that. Before we do that, though, we want to make sure that we know the right dose, the right titration, the right timing, all the minutiae of clinical development that you have to get right in order to maximize the effect of your drug. But I absolutely, I completely support that idea.
Thank you. I don't think we ever heard Russ recommend a head-to-head trial when he was working for the FDA. Our next speaker is Dr. Joe D'Souza from UCB to provide us an update on Brivaracetam.
Good afternoon. Thank you, Steve, Jackie, and members of the organizing committee for inviting me to speak and give an update on Brivaracetam. Here, my disclosure. Please note that Brivaracetam is an investigational drug and not approved by regulatory authorities, including the FDA. This is a summary of the key non-clinical features of Brivaracetam. And Brivaracetam is a compound from the UCB drug discovery program led by Henrik Klitgaard, and targeting to optimize binding to the SV2A ligand. So you can see that Brivaracetam has a high affinity and selectivity for SV2. And because of this high selectivity, it has little effect on other mechanisms, including activity on high voltage activated calcium channels and AMPA receptors. It has high lipophilicity, and due to that, it has high rapid blood-brain barrier uptake, and also a quick onset in animal models. In vitro, it has activity in drug-resistant spontaneous epileptiform activity, and very profound activity against the hypersensitivity in the high potassium, low calcium fluid model. In vivo, it causes potent and complete suppression of seizure activity in animal models of partial and generalized seizures. And at high doses is effective in the MES and PTZ models, which, as you know, were used for a long time to screen anti-epileptic drugs. Brivaracetam has a high therapeutic index at doses causing seizure protection and acute motor side effects. Here's a summary of the PK profile of Brivaracetam. It has rapid absorption and complete absorption throughout the GI tract. It has dose proportional pharmacokinetics from 100 to 600 milligrams, and its absorption is not affected by food. Protein binding is less than 20%, and the volume-based distribution is that of total body water, so you don't get any sequestering into tissues or major organs. Metabolism is primarily by non-cytochrome-dependent hydrolysis, and secondarily by oxidation mediated by CpCY2C19. 90% of the dose, 9% as parent drug, is excreted in the urine within 72 hours, and the half-life in healthy male adults is 8 hours. So far, there's no impairment of efficacy of the oral contraceptive at doses of Briv up to 400 milligrams. So this slide shows the 2 fixed-dose completed studies, and this has been presented before. The efficacy of the completed 2 phase 3 double-blind fixed-dose studies. Study N0153 achieved statistical significance for the primary efficacy variable, which was percentage reduction over placebo in the baseline adjusted partial onset seizures per week over the 12-week period of the study. And so was the 50% responder rates and the median percent reduction. N01252 did not meet the primary efficacy variable at the 50 milligram dose. However, statistical significance at a nominal value of less than 0.05 was achieved at the 100 milligram dose, and also for the 50% and median percent reduction. This slide summarizes the most common adverse events, seen as common events reported in the database that we have so far from all 5 double-blind placebo-controlled trials. You can see that we have over 1,200 patients in the Briv group and over 400 patients in the placebo group. The most common adverse events were headache, somnolence, dizziness, and fatigue. and the incidences were very similar to placebo. And there were no incidences in the Briv group that was twice as more often as the placebo group. This slide here summarizes, again, from the same database, all 5 double-blind placebo-controlled trials, the dropouts due to adverse events. And on the left are the 5 trials. The first 2 are the phase 2 trials. The second are the fixed-dose phase 3 trials. And the third one is a flexible-dose phase 3 trial. And on the right are the doses that were used in these studies. As you can see, in total, the dropouts due to adverse events was 3.9% for the placebo group and 5.3% for the Brivaracetam group. So the clinical trial program so far, we have in our completed trials, we have 32 completed phase 1 trials and 8 trials in phase 2 and 3 in adults and children with partial onset seizures and generalized seizures. This accounts to an exposure of about 2,000 patients with about 6,000 patient years of experience. And some patients have, because of the long development program, have continued on Briv for over 8 years. So I'd like to report today that we have just completed our phase 3 pivotal trial. This took us 3 years. We had to screen over 1,000 patients to get 760 patients worldwide. It was a randomized, double-blind, placebo-controlled, paragroup, multi-center study in focal onset and uncontrolled patients with focal seizures. So this is a 3-arm study comparing Briv 100 milligrams and 200 milligrams fixed doses with no titration. It was powered for both the FDA and the EU efficacy variables, which is median % reduction and percentage responder rates. So this is the largest study completed so far in partial onset seizures. We randomized 678 patients in 27 countries in the US, EU, and rest of the world with 208 active sites. So the study has been, enrollment has been completed. We've just, the last patient, last visit has just completed the study. So the results will be available in the second half of this year. Thank you very much.
Any questions for Dr. D'Souza? Chris?
Hi, Dr. D'Souza. That was really pretty good. I just question about the mechanism of brivaracetam versus levetiracetam in terms of how it binds to the SV2A. I know she mentioned it's more lipophilic, but if you can go in more detail how it really differs from levetiracetam in terms of its primary mechanism.
Well, I wasn't able to show the data comparing Lev and Briv in this study, but it does have a high affinity. It binds 10 times more strongly to the SV2A. It has high, compared to levetiracetam, it gets absorbed into the brain much quicker in rats, and now there'll be an abstract at AES to show a PET study showing the absorption, the quick absorption in Lev compared to brivaracetam in monkeys.
So the Tmax is only a half hour, so it's immediately absorbed.
It's almost immediately, yes.
Okay. Great. Okay, well, very good. Thank you very much.
Thank you, Chris. Please.
Thank you for the talk, Dr. D'Souza. One quick question. What would you expect the target recommended dose to be for brivaracetam?
Well, we would recommend the dose to be 50 to 200 milligrams, depending on the results of the last study. Thank you.
Great. Thank you very much. Our next speaker is Dr. Jackie French to speak about Vertex Pharmaceuticals VX-765.
So I think most of you know, and if you don't, I'm here to tell you that this drug is currently not in development, but we're talking about it anyway, okay, for anybody who's interested. Because there's been a lot of discussion about inflammation and anti-inflammatory therapies over the course of the last couple of days, and this is the only drug that actually has been in the clinic that the mechanism presumably is anti-inflammatory. So as you have heard, animal and human data from resected tissue strongly implicates cytokines and inflammation, not only in the development of the epileptic focus, but also in the perpetuation of the epileptic focus, and we've heard several people talk about that. VX765 was developed by Vertex, which really was not a CNS company at all. They were developing it for psoriasis, and then, several members of the epilepsy community came up to them and said, would you consider doing some trials in epilepsy and seeing if it would work? And the drug is an interleukin converting enzyme inhibitor, so it works by modulation of pro-inflammatory cytokines. Now, the person that was most interested in this drug from the get-go is Anna Maria Vanzani, who works at the Mario Negri Institute in Italy, and she had performed this particular animal experiment prior to us going to Vertex, and it actually uses the same model as we heard about yesterday that the NIH has now adopted as one of their interesting models, where you give hippocampal kainate, the animal goes into status, then there is a period where there are no seizures, and then eventually the animals develop stable epileptic discharges that look like mini subclinical seizures, and they're easily countable. So this is, as you heard, a very treatment-resistant model, and the animals don't respond to most typical anti-epileptic drugs, and this was a study, you can see the days at the bottom, it wasn't a long study, where you count baseline seizure rate, and then VX765, the compound I'm about to talk about, was administered in these chronically seizing animals, and you saw a slightly delayed, but not too much, but by about a day and a half, response with a significant reduction in seizures that continued while VX765 was administered, and then at the end of the study, in the last few days, days 8, 9, and 10, VX765 was withdrawn and the seizures recurred. So it wasn't, at least in their hands, it wasn't disease modifying, but it was a very short experiment, and perhaps if it had been given for more days, it would have been disease modifying. But that clearly was a very exciting thing for us, looking at this data and thinking, could this be a drug that would be useful in people with chronic epilepsy? And so the first study, again, a company that was, modestly interested in what their drug would do in the CNS, so we had a very small proof of concept study to see whether we were on the right track or not. This consisted of a screening period, then a 6-week baseline, and the one thing that we required for this study was patients with a very high seizure frequency, because at that time the company only had enough tox to cover 6 weeks of treatment, and therefore you had to have a lot of seizures in the baseline. So it was 1 seizure per week during at least 3 of the 6 weeks, and at least 6 total observable seizures, so 1 seizure a week overall during the baseline period. The other interesting thing about this study is that it allowed up to 4, not up to 3, but up to 4 background anti-epileptic drugs, because this wasn't the typical type of anti-epileptic drug that would tend to cause sleepiness, dizziness, and other things that would have a pharmacodynamic addition to the side effects of people's drugs. So that actually made recruitment quite easy. But as you can see, there were 48 people in the active arm and only 12 on placebo, so it was not powered to be a definitive study. Another thing that we wanted to know is whether there was any kind of disease-modifying effect, so we kept the drug going we kept the patients counting seizures for 6 weeks after we stopped the drug, just to see whether the seizures would go back to baseline or not. So they continued their seizure diaries. So let me talk about the safety first. It was very well tolerated. The adverse event rate was similar for the drug versus placebo. 1 subject on drug discontinued due to rash. And the serious adverse events were probably, they happened to be all on VX765, but I don't think we can blame VX765 for a hernia surgery. There was somebody who had some pain, and that seemed unrelated to drug, and 1 person had acute worsening of complex partial seizures. And that was, in fact, after the drug was withdrawn. So here are the results, so interpret them as you will. The first column shows you the typical results as we've been seeing from all the studies that have been presented, where the treatment period, there was an 8.6% reduction when placebo was subtracted. The 50% responder rate was 10% when placebo was subtracted, and the percent seizure-free was 12.5%. Oh, I'm going to talk about that in a second. Okay, so that was the treatment period with, again, placebo, and there was a very small placebo group subtracted. So that didn't look very encouraging. But we were kind of looking over, just eyeballing the data, and it seemed like the first few weeks that they were on the drug, they weren't doing that well, but as the time progressed, the people who were on drug and not the people who were on placebo seemed to do better and better as time went by. And we remembered that in the rats who had not been treated for very long, there also seemed to be a delay between when they first got the drug and when they got the results. So we thought, well, it's only 6 weeks, but let's just look at the last 2 weeks and let's look at the last 4 weeks. And then let's look at after the drug was discontinued to see if there's sort of a delayed response. Now, if you think about it, you think that if there's a placebo effect and it's psychological, after I tell you that the drug's been discontinued, you probably are not going to, have a placebo effect, I presume. But in fact, you could see that, again, the numbers look much better when you get to the last 2 weeks of treatment, and actually in a hybrid period that we put together. This is like post hoc of the last 2 weeks of treatment and the first 2 weeks of post-treatment, that actually the results look better. And you can see that, remember, these were people who were having at least a seizure a week. Most of them were having many more seizures than that. A lot of them actually had no seizures for the 4-week period of the last 2 weeks of treatment and the first 2 weeks of follow-up. So that was a little more encouraging, but it certainly was not proof of any sort, as you can see. But, we kind of had this feeling that we might be seeing a shift to the right of the effect of the drug, so a delayed effect that persisted into the post-treatment period, and then by the end of the post-treatment period, they actually went back to their typical 1 seizure a week or more. And that led us to design a second study. Now, I will say that I begged and pleaded with Vertex to do a small, definitive proof of concept study that would prove once and for all whether this drug would work or not. But for reasons that only people in this room who develop drugs might know, they decided that instead they were going to do the study to end all studies, which meant that there were 5, count them, 5 different arms to this study. So you can see there was placebo, 300 3 times a day, 600 3 times a day, 900 3 times a day, and 1,200 3 times a day. And each of these groups was going to have 100 patients in it, which would have been a very, very large study by anybody's standards. And unfortunately, after about 50 people or 60 people were enrolled in the study, the company decided, this is way too expensive a study. It's much too big a risk. We're not going to be doing this. Goodbye. Sorry, Vertex, if you're watching. But we appreciate everything that you did for us up until this point. We really do. As it turns out, as you can see, there were about 10 to 12 people per group, which, particularly if we're not talking about the lower doses being effective, is not sufficient patients per group to, again, have any definitive answer. So here's, this was presented at the AES last year. You can interpret it as you will. You can see the change in seizure frequency from baseline for the 300, 600, and particularly the 1,200 milligram group looked pretty good. And for some reason, the 900 milligram group looked bad. But I mean, with 10 people per group, and there were a couple of major outliers in that group in terms of very, very high seizure frequencies. it's not going to tell you very much. So what am I telling you? We still don't have the answer of whether an anti-inflammatory drug is effective for people with epilepsy. There's more data that we're going to be mining in this study. We wish we could have recruited some more people. And we also have many of these people remained on drug in a long-term extension for up to a year, and we're going to be looking at that data as well in the future. So the second study was terminated before a definitive answer could be obtained, and the question of whether an anti-inflammatory drug could be effective in treatment-resistant epilepsy remains open. Thank you.
Thank you. Do we have any questions? Steve, please.
So, Jackie, in a post analysis, I'm curious whether if you looked at the seizure frequency, say, in that 900 group versus the 1200 group, you know, to see whether maybe they had a lower seizure frequency, therefore less inflammation, or I guess what I'm getting at is the seizures presumably are releasing the cytokines through which, you know, 765 might be acting.
I do know that several people in the 900 milligram group actually had very high seizure frequencies. So that's not the answer. But, you know, we all know that neuroinflammation, you know, some people think MTLE, but in fact, if you look at the resected tissue of pretty much any kind of lesional epilepsy, you're going to see inflammation and probably non-lesional epilepsy as well. Whether some people, as with every therapy, are better candidates than others, we just don't know the answer. So we wish we had had more patients that we would be able to review, but this data will be reviewed and maybe we'll find a few answers. But it's all going to be very mined data at this point.
Nick, please.
Hi, Nick Poulos, University of Washington. Jackie, do you think there would be any advantage with this drug to taking a step back to the preclinical phase and maybe subjecting it to an ASP-like battery of differing animal models, maybe to find, you know, do some sort of dose ranging kinds of experiments and get an idea of, you know, where the efficacy in animal models might top out?
I mean, Steve should stand up again, but I mean, the only new animal model that would be interesting to put it in would be the murine encephalitis, but the acute animal models, MES, PTZ, 6 Hertz, there's absolutely no reason why it would work in those models.
Well, there are other animal models of, you know, spontaneous chronic seizures that generate, you know, clinical seizures that, you know, might be interesting to see.
You know, Anna Maria has used this molecule in some really interesting seizure models. So the one that just blows me away is that she used it in the Geyer's rat, and it reduced seizures in the Geyer's rat. No lesion, no, you know, KN8, nothing, no blood-brain barrier disruption. So that, to me, I think is the most interesting thing. So it is effective, and I'm not sure you're going to find a dose from looking at animal models that's going to be the human efficacious dose. But I think that this is a really interesting molecule, and hopefully it has not seen its last day. And I just want to say one other thing, and that is that the, you know, Vertex, thank you Vertex, have given us all of the data from these studies. They were done with the Epilepsy Study Consortium, and they have told us that we can have all of the data. And if anybody wants to help us look at that data, I would welcome any help that I can get. Thank you.
Thank you. Next speaker comes to us from Italy, Dr. Dorothy Giannetti, who will speak to us about Pitolisant.
Yes, ladies and gentlemen, Chairman, thank you very much for inviting me to present to you the data of the antihistamine 3 receptor antagonist, Pitolisant, and I will show you the data. Everything was financed by Bioprojet, a company in Paris, France. So the H3 receptor, it was in the 80s originally described as a presynaptic autoreceptor that inhibits the release of histamine in the brain. It also inhibits the release of other neurotransmitters. Here it is shown. It is found in human brain regions with high expression in prefrontal cortex, hippocampus, and hypothalamus. And it is very well found in the limbic system. So what about then the antagonists? The H1 and H2 receptor antagonists have been very successful therapeutic agents, and they were used in allergy and in gastric ulcers. So the H3 receptor antagonists are not yet on the market, but they are promised to be interesting for enhancing a variety of functions such as alertness, learning, memory, and Pitolisant has been studied also. There are publications, you can see phase 1 and phase 2 studies, and it has shown to improve refractory daytime sleepiness in patients with comorbid neurological diseases. But why would epilepsy be interesting for as a target? So there are case reports of seizures after use of H1 and H2 receptor antagonists, both in elderly and in children. And a study has been done in children with febrile seizures, and they found a low histamine concentration. And of course, mechanistically, it would make sense if it modulates activity of neurotransmitters like dopamine, glutamate, and GABA, that it could and should probably work in epilepsy. So we're talking about the Pitolisant, a non-imidazole-based inverse agonist antagonist. And it was studied in rat models, was effective in the GARS model, and specifically also effective in the kinase-induced epilepsy model and in the maximum electroshock. So a pretty wide range. Healthy volunteer data were gathered, of course, and up to 120 milligram, there was a Tmax between 1 and 3, and a clear dose-response relationship. So based on that, it was decided to perform a proof-of-concept study in epilepsy patients with 3 dosages, 20, 40, and 60 milligrams. So this has been published in Epilepsy and Behavior last year. And this is a collaborative study with my colleagues in France, from Paris, from Rouen, and from Marseille and Strasbourg. And that is Parrain, Janton, Masnou, and Edouard Hirsch. So we're talking about a phase 2A study with as main outcome, what is the goal? Suppression of photocally-induced EEG, epileptiform discharges in epilepsy patients. So here is an example of a patient. First, there is a placebo day, day 1 baseline, and photic stimulation. You see the lead down under. Can be performed regularly at different time points, standardized time points. So first in a baseline day. Then these data are collected in a way that the patients don't get seizures. You determine threshold sensitivity in 3 different eye conditions, and you end up with then ranges which can be transformed into a standardized photoparoxysmal or photoconvulsive response. So in this way, you get a lot of EEG information. After the second day, in which you give a single oral dose of the drug, you have a placebo day 3 just to look whether the effect of the drug might outlast several hours, as was the case, for example, in Brivaracetam. So these are the baseline characteristics of the trial participants. 14 patients were studied. There were the majority females, all but 1 had generalized tonic-clonic seizures. 5 were without co-medication. Spontaneous discharges were seen in 6 different subjects. Frontal epileptiform discharges are more over the occipital area, as well as 2 generalized. And here are the PPR ranges per eye condition, per patient. So here are the results. You can read this in detail in the publication. So the 3 dosages were tested. There was a clear dose-response relationship. Abolition of the PPRs was found more often at a higher dose, and also the reduction changes of, as is our standard, of at least 3 was also increased with increasing dosages. And which is a side information that also myoclonic jerks in patients who showed that clearly during photic stimulation were abolished. This is an example of EEG of one of the patients with the effect, same hour, day 1 versus day 2, so post-dose, and you see a clear difference. Time of onset was 2 hours post-dose, duration of effect 4 hours. And so graphically, these are the overall results. 9 of the 14 patients, so 64%, had either complete abolishment or significant suppression of generalized epileptiform discharges. And there was a dose dependency with strongest effect at highest dose. So to put a little bit things into perspective, I want to give you some information about this photoparoxysmal EEG response. You can find it at all ages, starting from age 2 till 60 years of age as well, more often seen in females. There is a clear relationship with seizures in daily life, so with epilepsy, and a variety of seizure types. It can be found in 40% in the easy-to-treat epilepsies, the idiopathic generalized epilepsies, and you can see it's hyperventilation as well as photic stimulation that can be used as a lowering of the threshold of these patients. Then you get an example, so both in absence seizures as in juvenile myoclonic epilepsy. This can be seen here are examples. Then also it can be found at basically the same percentage in patients with Dravet syndrome, and this is in the literature. And just recently we finished in Holland a study, and we found 43% in the Netherlands, apart from all other provocative factors that are well known in this type of patient. So this is an example of a patient with Dravet syndrome who showed a response. You find it in occipital epilepsies. This is a collection of patients otherwise completely normal with occipital seizures having visual aura, or basically these are seizures, symptoms, and also an example of the EEG. So that is the benign form versus the catastrophic form is the Lafora disease, where you can find it as well. And it's not so surprising that you can find it in the temporal lobe. So to finish my talk, you can even find it in patients with Alzheimer's disease. We found it just in Italy in 2 patients. So you can find it in all over. So I do hope this this Pitolisant find its way to like Brivaracetam and for the use of treatment in epilepsy patients. Thank you.
Thank you very much. Question, please.
Yes. So H3 antagonists are also known to have profound effects in terms of sleep architecture and arousal. So help me understand whether any of the subjects in your study reported changes in sleep quality, attention.
Actually, this wasn't studied. It is a single dose study. And of course, patients can show some side effects, and the side effect profile was not different from any other drug. But you're absolutely right; that needs to be done.
All right, thank you.
Good. Thank you very much. And now Dr. Ton Bund from IzumiBiosciences will talk to us about Elacridar, if I'm pronouncing that right.
Good morning. Thank you for the invitation to present. It's an honor with such an illustrious company here. I'd like to start with a riddle to shake it up a little bit. That is, if a patient with epilepsy, perhaps a child with epilepsy, starts responding to their drug.
Just get a little closer to the microphone, please.
I'm sorry, are we not?
No, you are, but just get a little closer.
Get a little closer.
Shouldn't touch it.
Okay, where's the, is there a control as well for the slides? Oh, there it is. Okay. My apologies for the false start. Nobody wants to miss the riddle. That's a good sign. We'll do a little quiz afterwards. So how come that if a patient with epilepsy starts off responding very well to their treatment, that over time they develop resistance to the same drug? What's the issue here? What's the riddle? Any answers? Any takers? Okay, let's move forward. We've heard a lot of beautiful stories about marketed drugs, marketed diagnostic tools, interventional tools, different tools in development, and what we'd like to discuss with you today is something different, what we call an oral pharmacoenhancer. It's not a drug, it's given concomitantly with other drugs to make them better, to actually enhance the penetration into the epileptic lesions and to restore therapeutic drug levels so that now the patient can actually respond again. So we're developing an oral pharmacoenhancer, and we're targeting the 30 or 40% or so of patients with epilepsy that indeed stop responding to their drug. So what are some of the reasons? Well, first of all, what's been mentioned is the blood-brain barrier as a major impediment to drug distribution into the brains. And in fact, it's a barrier for 98% of all of the FDA-approved small molecule drugs. Happily, the anti-epileptic drugs are all selected on the basis for their ability to cross the blood-brain barrier. But then over time, the same gatekeepers that are part and parcel of the blood-brain barrier also are upregulated in the epileptic foci, and so they in fact cause drug resistance. What I'll share with you are 3 rat models that have been developed independently from Izumi that show that a combination of known anti-epileptic drugs with an ABC inhibitor can in fact restore therapeutic drug levels and responses in these rats. About Elacridar, we know what the mechanism is. It's a potent, importantly equipotent, selective inhibitor of 2 gatekeepers or transporters, ABCB1, known as PGP or P-glycoprotein, and ABCG2, also known as BCRP or breast cancer resistance protein. And it's important to note that these are present in the blood-brain barrier. They can be upregulated, but they also function synergistically, so they're formidable gatekeepers. We'll share with you some of the pharmacokinetics that in fact you can give this together with anti-epileptic drugs. You can retain stable plasma concentrations of the drugs. You can maintain steady levels in the brain, but you really target the distribution to the epileptic lesions, which holds a lot of promise for a good safety profile. We'll share with you some of the pharmacodynamics, that in fact a number of ABC substrate drugs can now be targeted for increased therapeutic concentrations in these lesions. We'll go over the efficacy models. And in fact, we're starting with the Department of Defense on a funded project to enhance and create the next generation antidotes for poisoning with nerve agents. You may recall that in Syria last year, they used sarin to just wipe out entire villages. We're now working with the Department of Defense to create better antidotes to protect those victims. Previously, Elacridar has been taken into clinical development in 4 phase 1 clinical studies. It showed good safety, but it had horrible pharmacokinetics, and so that's the angle for us to really improve upon. What's interesting is that there's a strong regulatory precedent in FDA-approved pharmacoenhancers that you may know from heart therapy that are used in patients with HIV/AIDS. In fact, those pharmacoenhancers have a different mode of action in that they interfere and they slow down the metabolism, particularly of protease inhibitor, and thereby they increase the area under the curve, the Cmax, and importantly the Cmin. So now you get a nice buffer of the concentration at trough over the minimum effective concentration that's required for antiviral therapy. These products have been the basis of accelerated approvals with the FDA. They've become significant clinical and commercial successes, so I think it sustains the test of differentiation. And so we are developing, again, a pharmacoenhancer that has no inherent drug-like activity. So we like to partner with companies that have these drugs and that would like to develop it for this targeted patient population that have drug-resistant epilepsy. So maybe the most important part is actually the little visual on the bottom, where you see an anti-epileptic drug in blue that is present in the dark red plasma or blood compartment, and it travels across the blood-brain barrier. It passively diffuses into the brain. It's then recognized as a substrate by, in fact, two transporters. Here is only one depicted, PGP or P-glycoprotein. That's an energy-driven vacuum pump that readily vacuums out the drug away from the brain. It says, it doesn't belong here, I'm going to throw it out. And so, in effect, this pump is so effective that there is no therapeutic concentration, and if at all, it just whips in and out an insufficient mean residence time. So what we propose is that we can give the same anti-epileptic drug in combination with our pharmacoenhancer Elacridar. Now they travel across the blood-brain barrier together, but Elacridar has a much higher affinity for these transporters, and so it reversibly blocks the vacuum pump, and now we obtain the therapeutic concentration and the mean residence time to restore the efficacy in that patient with epilepsy. So here you see some data developed largely in the Netherlands, where you see that in patients with epilepsy, on the top left, you see the upregulation of, in fact, 3 transporters. What's important is that there's a very high baseline of BCRP or ABCG2 already in the human brain, and that there's indeed further upregulation. There's a dramatic upregulation in PGP from a very low baseline, so people like to talk about it. It's easy to measure. And then there's also an upregulated in MRP. On the bottom left, that you see in patients with brain cancer, that they much more readily upregulate BCRP, already from a very high baseline. And so those patients with brain tumors and epilepsy are highly drug resistant. What's very important on the right, most of the information about the functionality of these transporters shows, in this case from genetic knockout models that don't have any of these vacuum pumps, that these 2 pumps work together synergistically. 1 + 1 = 4. Or in fact, if you look at the case of imatinib or Gleevec, 1 + 1 = almost 13. So a very dramatic synergistic effect. On the next slide here, you see in 3 different rat models with different anti-epileptic drugs before and after that indeed we can restore efficacy. And now we get to, again, treat with an efficacious therapeutic level the same patient that previously was drug resistant. I should say these are rat studies, not patients. In the middle there, you see the pharmacokinetics, that indeed it is targeted distributions to the epileptic lesions, and there's a non-significant effect on the rest of the brain as well as the plasma levels. That's not been shown here. So here is the pharmacokinetics of 2 oral formulations that we developed that nicely obtained a concentration at trough in our target range between 400 to 600 nanograms per ml in a highly predictive rat model. Here we summarize that the improved PK and improved pharmacodynamics that I've shown you also translates into improved efficacy. On the top right, you see a model in adult glioblastoma in the mouse, where only the addition of Elacridar significantly improves survival. There's a similar publication in Press from Duke about a pediatric brainstem glioma model. In the middle, you see the basis for our contract now that we start with the Department of Defense, where in the case of a neurotoxin, there's improved neuroprotection by adding an ABC inhibitor and reducing significantly the convulsions. On the bottom right, we've already gone over the effect in drug-resistant epilepsy models. So in summary, we're ready to restart the IND with new anti-epileptic drugs, and hopefully together we can restore the responses that patients have seen previously on their anti-epileptic drugs. Thank you very much.
Thank you. In the interest of time, I think we'll move on, and Dr. Brunt will be available for questions afterwards. Our next speaker is Rick Stewart from Brabant Pharma to talk to us about Fenfluramine for Dravet syndrome.
Well, thank you very much for inviting us to present. We're here today to talk about 23 years of data in the treatment of Dravet patients from Belgium. So, as we've all heard over the last couple of days, Dravet syndrome is a catastrophic form of epilepsy with very few treatment options. We have been using Fenfluramine, as I said, for over 23 years, and it's the only drug that has actually demonstrated long-term seizure freedom in these patients. On average, we're looking at around about 6 years of seizure freedom with an average treatment period of over 13 years. The standard kind of response that we've been seeing in our last data point was that 92% of patients were either seizure-free or had a greater than 75% reduction in seizures. We've met with the FDA and the European agencies, and they've given us specific advice with respect to the trial design, and we expect to commence 2 phase 3 clinical trials towards the end of this year. We've got orphan designation granted in Europe and the US, and we're pending in Japan. And as with many other potential treatments for Dravet syndrome, we believe we've got potential for breakthrough status and other routes forward. So very briefly, we believe this is a transformational treatment. Our focus so far has been on seizure freedom. And we conducted a recent survey with the Dravet Syndrome Foundation, which identified that 3/4 of parents are looking for either seizure freedom or a greater than 75% reduction in seizures. And so far, no drug has actually shown that kind of response. The drug can potentially have significant benefits for patients, not only in terms of seizure freedom, the absence of status epilepticus can have a significant impact not only on the patient welfare, but also in terms of the pharmacoeconomic benefit to payers. And we believe that there is a potential for the reduction in cognitive impairment and the potential for a reduction in overall Dravet syndrome mortality. I'm going to cover here the history of the treatment with using Fenfluramine. It first started in 1998, and the most recent data point that we have is in 2014. The first report was actually in a publication by Boll and Caesar back in 1996, which examined the response of 11 patients taking Fenfluramine over a period of around about 8 years. This was subsequently followed up by a publication in Epilepsy by Lieven Lagai and Burton Kuhleman, which again identified the overall patient response. This was a group of 12 patients, and since then there have been public reports at the European Pediatric Neurology Society meeting in 2013, and most recently at the World Child Neurology Congress in Brazil. This is a uniform and consistent response that we're seeing over a very long period of time. But most recently, Brabant decided to have a more detailed examination of the data, and we conducted a longitudinal study by looking at each individual patient before treatment with low-dose fenfluramine, during every single assessment that they had over the period of treatment up until early 2014. And what we saw here was an even stronger response from the patients, particularly with respect to seizure freedom and the reduction in overall seizures. And what was important was it actually gave a very strong contrast between the Takayama publication in early 2014 and the response that we were seeing from our patient population. One of the important factors is, given that this is fenfluramine, there were concerns with respect to its previous use as a weight loss drug, and our detailed analysis over the whole duration of treatment identified that there were no significant cardiac effects, principally because we're using a substantially reduced dose of fenfluramine. What I'd like to do here is to give you a comparison of two patients from that longitudinal study. The patient on the left is our oldest patient. She was originally treated back in 1988, has now been on drug for 26 years. And what we've done here is to provide a common currency, is to have an annualized rate of seizures at the beginning. And then, as you can see over time, but certainly on the left-hand side, this patient has gone from approximately 12 convulsive seizures a year down to a very low level. In fact, her last seizure was about 9 months ago. There are always breakout seizures related to fevers, but this is a substantial change and potentially transformative change in her life. The patient on the right, patient 2, is our most recent patient. She was first treated in 2011, is currently 3 years old, was first treated at age 14 months. Prior to treatment by fenfluramine, she had 25 convulsive seizures in a 9-month period with 11 hospitalizations, and her most recent seizure was around about 5 months ago. So you can see from this analysis that low-dose fenfluramine is having a profound effect on the lives of these patients. So in conclusion, what we see here over this long-term observational study is that we have an average seizure-free period of over 6 years, and that 92% of the patients within the study were either seizure-free or had a greater than 75% reduction in seizures. And this is a truly potentially transformative drug for Dravet's patients. Thank you very much.
Questions related? Yeah, please. Do we have any questions? Dr. Cloyd.
Do you envision that patients who might be candidates for fenfluramine would require some special workup, particularly a cardiovascular workup, in order to take the drug?
What we actually intend to do is to have a full EEG workup. We are designing a Dravet syndrome care program. So at each stage, we're anticipating there'll be an assessment every 6 months. So at each stage, they'll have an EEG. So yes, we're very carefully monitoring the overall cardiac effect. But so far, over 23 years, we haven't seen any.
And you may have mentioned, I didn't catch it, are you envisioning, you must be envisioning adequately controlled trials in the U.S.?
Absolutely.
Could you just say something about that?
Yeah, we're actually having mirror phase 3 studies. We're expecting to have one phase 3 study in Europe with a mirror study in the U.S., 40 patients in both trials.
To take place when?
We're anticipating towards the end of this year.
Thank you.
Thank you. Michelle?
Yes, 2 questions. One, could you comment on concomitant drugs that were used in the studies? And the second question is, with all this data, can you tell us what the outcomes are of the individuals who've been on the drug for so long? You alluded to an effect on development. Are these people developmentally better than other people that have not been on the drug?
I'll start on with the first one, because that's the easy one. Yeah, most of these patients are either on clobazam, valproate, topiramate, so it's a combination. This is an adjunctive therapy. So it's a broad range, and indeed in a couple of instances we've actually seen them, because it's European, using stiripentol. In terms of the cognitive impairment, we have no data to suggest that's the case. Our expectation is that if we're able to treat the children early enough, and our expectation would be really from 1 year upwards, we may just be able to have some impact on cognitive impairment. I mean, it's not really known whether the cognitive impairment is related to the continued hypoxia from the seizures, or is it a component of the disease progression?
Okay, thank you. John,
just curious, any understanding of the mechanism of action, given that it's a low dose that you're testing? Is this releasing serotonin, or do you have any thoughts on that?
It's not fully elucidated yet, but we believe it's a postsynaptic 5-HT1A agonist. But we're still doing additional work in that area. Please.
It's just a quick follow-up question to John's. Is there any reason to believe that this might be effective in other epilepsies besides Dravet?
That is a very good question, and that's one I admitted from the presentation. What we'd like to do is to focus on Dravet syndrome initially, but we believe there's a potential that it might actually work in Lennox-Gastaut and potentially Doe syndrome and other refractory epilepsies. But one step at a time. We've got some very, very good data here, and the conduct of the clinical trials is absolutely critical. But yes, we are going to explore that.
Great. Okay, well, thank you very much. And our last speaker for this morning's session is Paige Figge from the Realm of Caring Foundation to talk about Charlotte's Web, facts and future.
Hello. Let me just say thank you for having me here today. As a parent of a child with a catastrophic epilepsy, it's an honor to stand in front of a room full of people who've addressed this in their professional lives and dedicated your lives to studying epilepsy and furthering drug treatment. My presentation is going to be a little bit different, a lot different than what you've heard today at this conference. I'm here to share a little bit of my experience with a brand new treatment option. One that has become a hot topic very, very quickly. It's often misunderstood, both by the patients and the medical communities. My daughter is Charlotte Figge. She is a Dravet child, as you can see. She has a uniquely severe case of Dravet syndrome. So when she was 5, we ran out of options, and we began treating her with a cannabis-based oil. And in Colorado, where we live, it's legal. And so we got her started. We lab tested it. The strain of plant that we use is extremely high in cannabidiol, extremely low in THC. It's actually classified as a hemp supplement. And it stopped her 300 weekly tonic-clonic seizures down to 0 from the first dose. That was 2.5 years ago. So she is drug-free, 2.5 years drug-free. She is no longer relies on her wheelchair, her feeding tube, oxygen. She is non-medical, and she is 99.9% seizure-free. So this is one anecdotal story. We kind of waited about 1.5 years to come out with the media about this, and we had 40, 50 patients at the time, and this was in August, and we were seeing these miraculous results from this oil, and they renamed the oil Charlotte's Web. So the strain of plant is Charlotte's Web. The oil is what we do is we have a specific milligram per milliliter infusion. It's in an olive oil infusion, and we have a dosing that's extremely similar to a pharmaceutical model. It's lab tested several times. We third-party lab test it to ensure the accuracy, and the Realm of Caring Foundation is our nonprofit that was built around this oil, this Charlotte's Web strain. And so we support and advocate and creates support for the parents that are actually migrating to Colorado as medical refugees to come for this treatment. As somebody who's seen a viable brand new treatment option in real life and in the faces that we work with in Colorado, it's become somewhat frustrating to watch as the body of evidence grows, but remains in the realm of anecdotal. We have several retrospective case studies some were compiled by people in this room on CBD extracts. We still recognize the need for new treatment options and to undergo the right processes and move this realm of evidence-based medicine as quickly as possible into clinical trials. We have this media attention, this hype surrounding this, and so we were faced with a bit of a dilemma, as you probably well know. The inquiries from families that the doctors and the scientists are getting, these are patients that are out of time, they're out of options. We realize this is a difficult situation to be in, to have a parent in front of you who desperately wants to try this treatment, but the medical community still does not have the tools that come from controlled trials. So for the realm of caring, this dynamic is equally as frustrating. The press surrounding this issue has typically focused on the success stories, the miraculous success stories, like Charlotte and some of the other patients that we have, many of the patients. This creates an unmanageable expectation. So the parents expect that 100% seizure control or 99% seizure control. They're moving their family across the country, from different countries often, to come try this. And it's, we really need to push forward with our clinical trials, as we will do. So I think that patients come and they expect to find a 100% seizure control, but some actually do not respond at all. This is just a new seizure treatment. As you in this room understand very well, these families don't have time to wait for this due process of the FDA process when the treatment option is available right now and showing some validity and some level of efficacy. This is why our organization finds it imperative to continue to provide channels for access. At the same time, we push forward to quantify Charlotte's Web with the best possible clinical trials. The greatest hurdle to clinical trials in the United States is the scheduling of this substance. It's a Schedule I on the Controlled Substances Act. The plant is a Schedule I, the cannabinoid is a Schedule I, the CBD is non-psychoactive, and yet it's still a Schedule I substance. So fortunately, many other countries have invited us to come in and do our clinical trial in their country. So that's what we're doing for the immediate future. We're hopeful for the data that's going to be accumulated in the coming months from a blinded placebo-controlled trial. It will begin in Uruguay, and it'll likely end up as a multi-center trial utilizing institutions in several other countries as well. The trial protocol is in its final stages. It's being developed by the combined efforts of Dr. Orn Davinsky, Dr. Edward Ma, the advisory help of Dr. Roger Porter, and the preclinical efforts of Steve White. So that's in a quick nutshell. I know we ran over today, but I just wanted to come out and explain what is happening in the future with Charlotte's Web and what is happening now, and just kind of clear up some of the misconceptions. And I really want to thank you so much. The Epilepsy Foundation has been a really validating supporter for us behind this, for the parents who are using this and seeing results, real results. And so I just want to say thank you very much. I know this is kind of out of the typical norm for the presentation, so thank you for having me.
Thank you. I'm sure there'll be some questions from the audience.
No.
No. Maybe it was atypical, but I think, speaking for the audience, it was wonderful, and we really are very grateful for you being here today. Elson, please.
Question. Thank you very much for sharing your personal story. It's so important for us to hear about it. My only question is that if you should get the results from Uruguay, did you say, the country, is it something that would be acceptable to FDA, or do we have to jump the hoops again? Certainly, it will serve as a sort of motivation to the public and to FDA, to government agencies to do such studies here in the U.S. too. But what would be the status of that study for the United States?
I think the protocol is just slightly different down there. We're going to use the same protocol that is being used here. It's just that we're using the whole plant instead of isolating certain compounds in the plant. So there's hundreds and hundreds of things in the plant, and that's why it's a little bit different. I don't know how that's going to be viewed by the FDA, to be honest.
Great. Dr. Katz?
Yeah, well, just to follow up again, I haven't been involved in any of these discussions. I don't know if you have been discussing this with the FDA. There's no rule that says the agency couldn't rely on a study or even an entire data package, an entire NDA that's performed outside the United States. So that's possible, and it's happened. Of course, there are issues of data integrity, and every time a study is done in a place that the agency doesn't really have a lot of experience with, because they look very closely at the data and this sort of thing, but there's no a priori reason that couldn't be acceptable. I would say, though, based on your last comment, that the entity being studied is not exactly the same as the entity that you would presumably want to have marketed in the United States, that is likely to raise questions because there are many other compounds, obviously, in the plant than in just pure cannabidiol. So I would predict that that would be an issue. But the fact that it's done elsewhere doesn't have to be a big issue.
Oh, okay, thank you.
Well, thanks again. I want to thank all of our speakers. And one final question, please.
And any negative side effects? Have any kids regressed on?
What we're seeing is, and you know, I'm not a doctor, I'm a mother. What we're seeing and our doctors are watching is a possible interaction with other seizure drugs that the children are on. So the CBD alone, we're not seeing side effect from that, which is equally as exciting to have a new seizure treatment that has no negative side effects and actually helps with sleep, behavior, and appetite, the three very common detrimental side effects of typical seizure drugs. But no, we're not seeing that. We do see some possible interactions, levels raised in the blood of some other drugs that are metabolized in the liver on the same enzyme.
Great. Well, I'm sorry we ran over, but it was well worth the wait. And thanks again, Paige. And
thank you.